Good morning, everyone. The topic of my presentation today is Pbpk absorption modeling to support risk assessment and the Biowaiver for generic oral products. Here is my disclaimer. In this presentation, I will first talk about Pbpk absorption model and its applications, then highlights of recent oral PPK impacts on regulatory decision-making in OGD, followed by highlights of oral PPK research to support Biowaiver and risk assessment. This slide shows the general strategy of the establishment and application of Pbpk absorption modeling. As can be seen from the schematic figure, the model inputs include API-dependent parameters, formulation-dependent parameters, and physiological parameters. After oral dosing, drugs are absorbed through the GI tract and get into portal vein and systemic circulation, which links to drug exposure. In such a way, using the established and validated Pbpk model, the effect of critical quality attribute changes, for example, dissolution on in vivo performance, can be predicted through virtual BE simulations, and then to set clinically relevant specifications or to support BE evaluation. In the year of 2020, FDA issued 2 guidances supported by Pbpk regulatory applications and research. One guidance is the use of Pbpk analysis, biopharmaceutics applications for oral drug product development, manufacturing changes, and controls. Another guidance is evaluation of gastric pH-dependent drug interactions with acid-reducing agents. These 2 guidances describe the utility of Pbpk absorption modeling for biopharmaceutics applications as well as for predicting the pH-dependent DDI potential. This slide shows the regulatory questions that Pbpk absorption model can answer in generic drug development. They are all related to BE assessment, including setting dissolution safe space, evaluating the impact of changes in critical quality attribute, assessing the risk of formulation mechanism changes, etc. The Pbpk absorption modeling can also be used to predict the PPI, DDI potential, and GI local concentration, support BE assessment in specific populations, and the waiver of in vivo studies. This table summarizes the highlights of recent oral PPK impacts on regulatory decision-making in OGD related to risk assessment and Biowaiver. For example, Pbpk was used for evaluating the impact of particle size distribution on BE and support setting a clinically relevant 3-tier PSD specification. Pbpk was also used to evaluating the impact of faster dissolution profile of lower strength on in vivo bioequivalence and support Biowaiver. The case example I show here is about a capsule product for which the PK parameters were found to be sensitive to changes in mean particle size of drug X. The applicant submitted a mechanistic absorption model to link particle size distribution with in vivo PK data. FDA reviewers conducted reviews and simulations using Pbpk model with fixed D50 and changed D10 and D90. The results are shown in this table. The modeling simulation results suggested that with a certain fixed value of D50, the test versus reference PK matrix showed a low risk of non-BE when D90 changed over a wide range. In summary, these modeling results support a satisfactory BE assessment of this ANDA and setting a clinically relevant 3-tier PSD specification. The second case example is about using Pbpk absorption modeling to support waiver for lower strength with in vitro testing deviations. We know that waiver of lower strengths can be dependent on formulation proportionality, dissolution similarity, bioequivalence on other strength. However, this case example has dissimilar dissolution profiles for lower strength of the test product. Thus, Pbpk-PBBM modeling was used for predicting the impact of faster release of lower strength on the bioequivalence under fasting and fat conditions. After review, we identified some deficiencies with the submitted model, especially on the validations. When these deficiencies addressed, the developed Pbpk-PBBM model can be used in assessing the impact of dissolution differences on in vivo performance and bioequivalence. This slide shows the highlights of oral PBPK research to support Biowaver and the risk assessment, including GDUFA-funded research projects, expanding BCS Class 3 waivers for generic drugs to non-Q and Q2 products, and internal research best practices for using oral PBPK modeling to support generic drug development. Another area that Pbpk can be used is for supporting Biowaver. Currently, there are two main general guidances for BABE waivers based on BCS. One is the guidance for waiver of in vivo BABE studies for IR oral products based on BCS, which was issued in December 2017. The other one is ICH-M9 guideline for BCS-based biowaivers, which was endorsed in June 2018. Both guidances describe that for BCS Class 3 drug products, Biowaiver should be supported by high solubility of the drug substance, rapidly dissolving of drug product, and the test product formulation is qualitatively the same and quantitatively very similar. Biowaiver option is also available for BCS Class 3 generic drugs. For example, product-specific guidance for hydroxychloroquine sulfate oral tablet also includes BCS Class 3 based Biowaiver option. The language regarding the requirement is similar to what is described in previously mentioned two general guidances. For the formulation that is not Q1 the same and Q2 very similar, the impact of excipients on the permeability and in vivo performance would be very important. To fill this knowledge gap, OGD has funded a contract, namely expanding BCS Class 3 waivers for generic drugs to non-Q and Q2 products to assess the impact of excipients on the dissolution and permeation of BCS Class 3 drugs in solid oral dosage forms. The research results could increase the confidence of using varying amounts of excipients. In addition, to investigate the impact of excipients on the in vivo performance of BCS Class 3 drugs, Pbpk modeling may potentially be used as an alternative B approach and to support Biowaiver of non-Q and Q2 BCS Class 3 generic drugs. This case example here showed that FDA researchers developed Pbpk models and conducted parameter sensitivity analysis on the effect of changes in passive permeability on Cmax and AUC for representative BCS Class 3 IR drug products. It demonstrated the potential capability of Pbpk model to ascertain the potential impact of excipients on drug absorption and bioavailability. We also internally developed a Pbpk model for putative BCS Class 1-3 drug oseltamivir phosphate, OP, and its metabolite, oseltamivir carboxylate, OC, in both adults and pediatrics. Then conduct virtual BE simulations to establish BE dissolution safe space for OP in both adults and pediatrics. This case example not only highlights the utility of Pbpk absorption modeling and simulation for setting clinically relevant dissolution specifications in both adults and pediatric populations, but also demonstrates the potential utility of Pbpk absorption modeling as alternative BE approach to support Biowaiver of non-Q1Q2 BCS class 3 drugs. Details can be found in this AAPS journal paper listed here. When developing Pbpk absorption model for supporting risk assessment and Biowaiver, there are always some challenges but also opportunities for addressing those issues. For example, investigators is trying to establishing correlations between the in vitro characteristics and observed or estimated pharmacokinetics. To establish this relationship, dissolution method may need to be biopredictive. In addition, the modeling of in vitro dissolution profiles, the combination of drug formulation-specific properties with GI variability also need to be considered. In addition, GI physiology related to absorption of BCS class 3 drugs include permeation. The impact of the formulation excipients on permeation need to be considered as well. Also, the modelers need to inform the models with correct estimates of within subject variability, which can be obtained from previous replicated study or included in system parameters. As demonstrated above, we have conducted research related to oral PBPK modeling through both internal and external projects. Here listed a couple of recent publications based on these research projects. You may refer to these publications for details. In conclusion, with continuous improvement along with more submissions and research, PBPK absorption modeling would aid in setting clinically relevant specifications, conducting risk assessment on the impact of critical quality attributes, supporting high-impact applications such as alternative BE approach and Biowaiver, and evaluating the impact of excipients and food on bioequivalence. FDA encourages the sponsor to submit alternative BE proposals with modeling and simulation data. Early communications with agency where pre-IND meeting and controlled correspondence is encouraged. With that, I would like to thank all the people listed here for their support and hard work, especially the reviewers and scientific assessors involved in the regulatory work. Also, special thanks to those authors who are involved in the publications that I mentioned in the slides. This concludes my presentation, and I will be back later to answer questions during the panel discussion. Thank you.
Hello, my name is Kimberly Rains. I'm a supervisory pharmacologist at the FDA. I'm currently serving as a branch chief in the Division of Biopharmaceutics, Office of New Drug Products, Office of Pharmaceutical Quality. I would like to thank the organizing committee for the opportunity to speak in this workshop. My presentation is entitled The PBPK Biopharmaceutics Guidance and Progress on Risk Assessment. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. The Office of Pharmaceutical Quality believes a quality product of any kind consistently meets the expectation of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients the confidence in their next dose of medicine. For this presentation, I will be covering three main objectives. 1. To define and understand the drive to develop the PBPK biopharmaceutics guidance. 2. To comprehend the purpose and the framework of biopharmaceutics risk assessment. And 3. to demonstrate how in vitro dissolution can provide predictive insight to in vivo performance. Before we begin, I would like to provide some definitions to frame this discussion. Biopharmaceutics is the study of physical and chemical properties of a drug in its dosage form as related to onset, duration, and intensity of drug action. PBPK for biopharmaceutics applications, also known as PBPM, is defined as physiologically based pharmacokinetic absorption models including advanced compartmental absorption transit and advanced dissolution, absorption, and metabolism, as well as other mechanistic models, which mimic physiological conditions, incorporate dissolution information, while accounting for relevant physiochemical and physiological factors leading to prediction of systemic exposure versus time. And lastly, a term that I'll be using throughout this talk is patient-centric quality standards. And these are defined as a set of criteria and acceptance ranges to which drug products should conform in order to deliver therapeutic benefit indicated in the label. I like to think when you start something, you want to keep the end in mind. So what is the end in mind for the future state of dissolution testing? OPQ management has stated an in vitro dissolution test that provides predictive insights to in vivo performance. This ensures high quality drug products that maintain safety and efficacy throughout the product lifecycle. With a predictive dissolution, the impact of critical material attributes and critical process parameters on in vivo performance can be quantitatively assessed by the in vitro dissolution. This provides scientific and risk-based knowledge to support patient-centric quality standards. I've included a cartoon of a bridge at the bottom of this slide to show how we're bridging this scientific and risk-based knowledge to the patient-centric quality standards. This concept isn't new. It's something that's been one of the goals of the agency for quite a long time. All of the hard work that was put into the 1997 guidance, extended release, oral dosage forms, development, evaluation, and application of in vitro and in vivo correlation was really trying to bridge that gap between the in vitro and the in vivo performance. This thought process has maintained and continued in the Office of Pharmaceutical Quality now. It has led efforts for development of the guidance of dissolution testing and acceptance criteria for immediate release solid oral dosage form drug products containing high solubility drug substances, which went to standardize the dissolution testing would provide benefits to both industry and the agency, as well as the collaborative effects for the ICH M9 Biopharmaceutics classification system-based biowaivers. In October of 2020, the agency issued the draft guidance for the pharmaceutical industry, the use of physiologically based pharmacokinetic analyses, biopharmaceutics applications for oral drug product development, manufacturing changes, and controls. Physiologically based pharmacokinetic analyses combines physiological descriptors such as blood flow, tissue volume, partition coefficients, and population estimates, along with unique drug substance and drug product characteristics to model and simulate the pharmacokinetic and/or pharmacodynamic behaviors in vivo. This draft guidance offers several high-level workflows that include defining the objective of a PBPK model, model development including structure, assumptions, and parameterization, model validation, and the critical step of application of the model to its purpose. As noted, the guidance is only applicable for biopharmaceutics applications of orally administered drugs, and this will be the main focus of our talk today. There were many activities leading up to the publication of the guidance. The agency had held workshops, had performed collaborative research opportunities with industry and academia, as well as the agency was receiving a number of PBPK for biopharmaceutic assessment submissions. Briefly looking at the submissions that we were receiving, many of them contained PBPK to support dissolution specification setting, qualification of particle size distribution, as well as supporting risk assessment. We also experienced some use of Pbpk for biopharmaceutics assessment for SUPEC changes, scale-up and post-approval changes for both new drugs as well as NDA submissions. It is noted the impact of biopharmaceutics modeling is very large. It can be impactful in a number of different areas. Some of those areas include in assessing when BE studies are needed, to support IVIBC and IVIBR approaches, for setting in vitro dissolution specifications, as a tool for risk mitigation, for the development of dissolution models, as well as for applications in BCS and Pbpk-based biowaivers. Noting what Pbpk for biopharmaceutics applications can bring, we also note that there are a number of challenges with providing this information, including the absence of in vivo data available for model verification and the appropriate use of extrapolated information, the evolving knowledge on GI physiology, constraints of molecule validation, and the current lack of regulatory harmonization. We see those challenges as opportunities to move forward because we understand this important role that Pbpk for biopharmaceutics applications has. Going back to the definition of biopharmaceutics that I gave earlier, this is just a schematic showing how this drug product quality is related to the patient and how we can use this information to give us a science and risk-based approach for our patient-centric quality standards. So a clear understanding of the critical bioavailability attributes, dissolution included, and applying them to the in vivo performance of the drug product through bioavailability or bioequivalence, we can find this link in Pbpk through biopharmaceutics application. And this helps us determine a consistent therapeutic benefit for the patient. As shown in that previous slide, our understanding is based on this risk-based assessment. One of the drives in the agency moving forward is to really have a knowledge-aided structured assessment, building in that risk assessment. And here is the process by which we're doing it. It's known as CASA. CASA is an integrated set of tools and frameworks that aid in regulatory assessment and knowledge management. I'll focus on the KA portion. The KA is built of 3 pillars. Pillar 1 is the assessment of risk to quality. Pillar 2 is the control of risk by assessing the product. And Pillar 3 is control of risk by assessing manufacturing and facility. Herein is how we can build in this risk assessment into our assessments and leverage this information, for example, Pbpk for biopharmaceutics assessment. Built into the CASA framework is our initial biopharmaceutics risk mitigation strategies. The biopharmaceutics risk is identified by changes in dissolution that may impact bioavailability. We identified 5 qualitative risk levels, beginning with very low. Very low, its expectations are that dissolution would be rapid and there would be no impact on bioavailability. In these instances, a standardized dissolution test could be incorporated as per our high solubility drug substance dissolution guidance. For low risk, there's a little more of an impact, but we still expect immediate release products to behave with rapid dissolution. And in those instances, although not a standardized method, an adequate method development to justify dissolution method and acceptance criteria could be used. For those that have a medium risk, where there is more likelihood of an impact of dissolution and changes in bioavailability, an in vitro approach is used to mitigate the biopharmaceutics risk, and the dissolution test should target and to detect meaningful changes in identified critical bioavailability attributes to provide insight into in vivo performance. The next risk is high, and I've circled this here because this is where there is an impact to bioavailability, and we would expect that an in vitro in vivo relationship is used to support patient-centered dissolution tests, and this could be based on in vitro in vivo data and or PBBM, as our focus is here. For the very high risk, we would want in vivo studies to develop an IVIVC or IVIVR to support this patient-centric dissolution test. Now I'll give some examples and walk through some decision trees and how we make some of these determinations on the risk level. These are just presented for examples, and I'm starting off with products that are immediate release solid oral dosage forms, and this does not include any evaluations for narrow therapeutic index drug products or drug products that are required to have a rapid onset of action for their clinical benefit. The principles behind the risk assessment are based on the physiochemical properties of the drug substance and the drug product. We are not recreating the wheel, we're just using these characteristics to define our initial biopharmaceutics risk. We begin with the drug substance solubility, and is it high per the ICH-M9 or the BCS Biowaiver guidance? If they're able to demonstrate high solubility, again, we'll reference the guidance that was recently published in 2018, and we'll see if they can also achieve rapid dissolution under the standard conditions, which are 500 ml of 0.1 normal HCl in an aqueous medium. If they're able to achieve rapid dissolution, then that would be characterized as very low risk, and we can implement the standardized dissolution test and have little concerns because the biopharmaceutics risk is considered very low. However, if they're not able to meet this rapid dissolution, it can still be considered a low risk as shown in the slide before, but we would just have to justify the acceptability of their method and acceptance criteria. In instances when solubility is not high and it's low or unknown, then we would take to looking at the evaluation of the permeability. If the permeability is high, then we can address to see if the dissolution of the highest strength shows rapid dissolution in media with a pH range of 4.5 and 6.8 without surfactant. In these cases, we presume that the risk would also be low. These could be some of the weak acids that may be pH-dependent solubility. And as long as we ensure that the drug product is dissolved in the GI tract, there should be little concern. So we would characterize those as low risk. However, if they're unable to rapidly dissolve under those conditions, we would move forward to looking at if other critical bioavailability attributes could be clearly identified, detected, and controlled. And to find critical bioavailability attributes, or CBA, they're formulation or process variables that are expected to critically impact bioavailability, as in the rate of absorption rate and extent of a drug product. So these could be a number of different things: particle size, hardness, or number of characteristics related to the drug product or the manufacturing process. If these are identified and controlled, we can assume that it's a medium risk, and having all of these quality attributes controlled will help control the quality of the product. However, if we're unable to identify any of these CBAs, this becomes a high risk or a very high risk. And this is dependent on if an IVIVC or IVIVR can be established with PBBM or any other available in vitro or in vivo data. If we can use PBBM to support this, this would be a high risk. This is just the initial risk. So once this is developed, this does help mitigate the risk, and that goes into the risk assessment discussions that we spoke about earlier. However, if they're unable to use the PBBM approach or another supportive in vivo or in vitro data, then that would be characterized as a high risk, and we would expect in vivo data used to support setting up the dissolution method and acceptance criteria. Next, I'll move on to looking at extended release solid oral dosage forms. And again, this example decision tree does exclude narrow therapeutic index drug products. Here, the questions are a little similar, but based off of the characteristics known for an extended release product. The first question is the in vitro dissolution independent of test conditions, for example, the media pH, rotation speed? If dissolution is consistent and not pH and not dependent, then that would be a medium risk. And we proceed similarly as we did in the last decision tree by evaluating the CBAs to ensure that they are adequately controlled and identified. However, if the dissolution is not independent, and they're unable to identify a CBA, then we would move forward to looking at if an IVIVC can be established with PBM or any other available in vitro or in vivo data. Again, if this can be done, it's initially a high risk, but with the development of a PBM or development of a IVIVR, we could mitigate that risk. However, if they're unable to satisfy that, then that would make it a high risk. And then high risk would need to be supported by in vivo data. These decision trees hope to demonstrate how we're evaluating the initial risk related to in vitro dissolution. It is noted that we would expect that most of these products that we review in the Office of Pharmaceutical Quality may fall in the very low, low, medium, and sometimes high, and very infrequently very high categories. Now I'll walk us through a few case examples. Case example number 1 is for an immediate release tablet. It's indicated as an antipsychotic for the treatment of major depressive disorder and schizophrenia, but it is not a narrow therapeutic index drug. It's available in a number of strengths. It's classified as a BCS Class II drug substance per literature, and the formulation process is dry granulation. For reference, I've provided the solubility over the physiological pH range, and you can see that solubility is low and pH dependent. So now let's walk through the initial biopharmaceutics risk framework and answer the following questions. Is the drug substance solubility high per the BCS guidance? As demonstrated in the table on the previous slide, we would say no. The next question would be, is the drug substance permeability high? We could reference the label for this information or literature to see if there is data to support the permeability if it is not provided. Based off the label, it says that permeability is high with absolute BA of 95%. The next question would be, does the in vitro dissolution of the highest strength show rapid dissolution in media with pH range of 4.5 to 6.8? In the data that was provided within the submission, it showed that dissolution was not rapid in the range of 4.5 to 6.8. So that leads us to the next question. Can critical bioavailability attributes be clearly identified, detected, and controlled? Based off the information that was provided in the pharmaceutical development report, there were a number of critical bioavailability attributes identified as well as controlled within the drug product manufacturing and formulation process. Therefore, this would be identified as a medium risk. On this slide, I just show some dissolution data and show that the dissolution method was discriminatory for the API particle size, and we could assume that that is a CBA, and since it's controlled at the particle size level, they had a 3-tier specification. We were able to set an appropriate dissolution method for this drug product. Moving on to case example number 2. This is for an extended release tablet. It's indicated as an anti-epileptic, but again, it is not qualified as a narrow therapeutic index drug product. It's available in a number of strengths. Literature states that it's a BCS Class II drug substance. with pH-dependent solubility, and solubility decreases with an increase in pH. The formulation is extended release core coated with an enteric coating, and there is proportional similarity among the strengths. It's manufactured by dry granulation compaction and enteric coating. So let's walk through the decision tree. Is the in vitro dissolution independent of test conditions? When evaluating the dissolution data that was submitted as part of the pharmaceutical development report, it was shown that dissolution was pH dependent. So the next question would be, could critical bioavailability attributes be clearly identified? By evaluation of the pharmaceutical development report, there were a number of CBAs that were identified as well as controlled. Therefore, this would fall under the medium risk category. The applicant proposed the following dissolution, and it was shown to be discriminating against variations in the release controlling polymer, which is a critical quality attribute of this extended release formulation. The proposed dissolution criteria at the 6 and 11 hours is permissive, but they were able to use supportive bioequivalence data as a pilot study to support. Were they able to show that these two pilot batches were bioequivalent, even though the dissolution ranges greatly differ? Therefore, this supported a widening of the dissolution acceptance criteria at the 11-hour time point. I'll move on to case example number 3. This is an immediate release tablet. It's indicated to reduce the risk of stroke and systemic embolism in patients with non-valvular atrial fibrillation and for the treatment of deep vein thrombosis and pulmonary embolism. It's available in a number of strengths. The BCS class of the drug substance is considered class 4, low solubility and low permeability. The dissolution data contained in the submission was not discriminatory to changes in drug substance or product attributes, and the dissolution method did not reject a batch that was not bioequivalent to the phase 3 clinical batch. So walking through the decision tree, is the drug substance solubility high per BCS guidance? No. Is the drug substance permeability high? No. Could critical bioavailability attributes be clearly identified, detected, and controlled? No. Could an IVIVC or IVIVR be established with PBPM and/or any other available in vitro dissolution data? That's where we landed, and that's where the question was, and I'll give you a preview that the answer was yes. So this did fall into a high risk category. So for this application, they were able to use PBPM to mechanistically model and simulate for their IR product. Briefly walking through the model building, they used IV PK data, oral PK data at one specific strength. They were able to input physiochemical properties, absorption parameters, and PK parameters, as well as including dissolution profiles. Model validation, there was an internal validation with one strength, an external validation with another strength, and repeated virtual bioequivalence trials. And where were they able to show that one strength was not BE to the internal validation strength, and where the other formulation was determined to be BE. And they were also able to validate this model. And then they applied the model to support biopredictive nature of the new dissolution method that was identified and to set a dissolution specification. And here we see the model application. The generated virtual profile with a Q equals 75 and 30 minutes was run through virtual BE and shown to be bioequivalent. Therefore, the applicant was able to support the clinical relevance of their proposed Q equals 75 and 30 which differed from the agency's recommended Q equal to 80 in 30 minutes. In summary, I hope the following was learned through this presentation: that biopharmaceutics helps link pharmaceutical quality to the patient in a meaningful way and assure consistent drug product performance, that this framework allows for regulatory flexibility based off of risk rather than manufacturing capability, and the use of Pbpk for biopharmaceutics application in drug products demonstrating high initial biopharmaceutics risk is very useful. What's on the horizon? We hope an increase in the number of successful regulatory submissions for Pbpk for biopharmaceutics applications, continued collaborative research opportunities with industry, academia, and regulatory bodies, as well as data sharing to continue to grow the science. I would like to thank my colleagues in the Office of Pharmaceutical Quality and the Division of Biopharmaceutics, as well as my supervisors and management. Thank you for your time.
Hi, I'm Chris Bode from Absorption Systems, and I'm going to talk about some in vitro work that we've done to investigate the feasibility of expanding BCS Class 3 waivers for generic drugs to non-Q1, non-Q2 products. By way of disclaimer, I am not an FDA employee, and this presentation reflects my views and should not be construed to represent the views or policies of the FDA. So the first regulatory guidance on the BCS was issued by the FDA in the year 2000. It has been updated a few times, and parts of it have been adopted by other regulatory agencies. The harmonized ICH M9 guideline on the BCS went into effect in Europe in July 2020 and in the US in May of this year. In this guideline, and in recent versions of the FDA guidance, Class 3 biowaivers are allowed, but the eligibility criteria are extremely conservative in terms of formulation similarity for a generic versus the corresponding reference listed drug, or RLD. The generic formulation must be Q1 or qualitatively the same, meaning it must have all the same excipients, and it must be Q2 or quantitatively very similar. RLD formulations are proprietary, and for that and other reasons, in practice, it is very difficult to satisfy these criteria. So it should come as no surprise that the BCS biowaiver pathway has been vastly underutilized for drug products containing a Class 3 API. And this project is one approach to investigate the feasibility of expanding Class 3 biowaiver eligibility to include non-Q1, non-Q2 formulations. In addition, there's a need for a better in vitro model to evaluate the effects of excipients, and qualification of such a model was also part of this project. So the first aim was to qualify a proprietary model, the in vitro dissolution absorption system, or IDOS, for assessing excipient effects on drug permeation, and that's the focus of this talk. The second aim, which basically corresponds to the second year of the contract, is to look at the impact of different combinations of excipients in solid oral dose forms on drug dissolution and permeation measured simultaneously using IDOS. And this is IDOS. It's an in vitro system for simultaneously assessing the dissolution and permeation of solid formulated drug products, either tablets or capsules. IDOS consists of a 500 ml dissolution vessel containing 2 permeation chambers, 8 ml each, and at the interface between the dissolution chamber and each permeation chamber is a polarized monolayer of human intestinal Keiko2 cells. The apical side of the cell monolayer faces the dissolution chamber, containing a biorelevant buffer, in this case fasted state simulated intestinal fluid, or FASIF. The basolateral side of the cell monolayer faces the permeation chamber, which is the receiver side of the device, containing a buffer at pH 7.4 with serum albumin to mimic blood plasma. Dissolution of a drug product can be monitored by measuring the amount of dissolved API in a dissolution vessel over time, filtering the samples to remove undissolved API. And permeation across the Keiko-2 cell monolayers is assessed simultaneously by measuring its rate of appearance in the permeation chambers. First, we qualified an analytical method for 5 model drugs. These are the 5 drugs, and there was a reason for selecting each of them. Acyclovir, cimetidine, and ranitidine are class 3 drugs with clinical data on the effects of excipients on drug absorption. Atenolol is a class 3 drug that we included as a cell monolayer integrity marker, and the class 1 drug minoxidil was included with the expectation that its permeation would not be affected by excipients. We pre-dissolved the 5 model drugs and co-dosed them in IDOS as a cassette, without excipients and with 15 different excipients, one excipient at a time, each at 3 concentrations. But even though IDOS is capable of measuring dissolution and permeation simultaneously, in this phase of the project, the cassette of model drugs was pre-dissolved, so we're only looking at the effects of excipients on drug permeation. These are the 15 excipients that we tested and the 3 concentrations of each. In general, with a few exceptions, the high test concentration is equal to the highest amount of a given excipient in an immediate release solid oral dose form dissolved in 250 ml, and the amounts come from the FDA's inactive ingredients database. The mid concentration is generally 25% of the high, and the low concentration is generally 25% of the mid. The 15 test excipients were selected to cover all of the major functional classes of excipients, from fillers to disintegrants to lubricants, and 1 surfactant, sodium lauryl sulfate, or SLS. In each experiment, there was a negative control with 2 dissolution vessels, 4 permeation chambers, a positive control with 1 dissolution vessel, 2 permeation chambers, and 3 concentrations of an excipient with 3 dissolution vessels and 6 permeation chambers. Note that this is an in vitro system, and the receiver concentration of the model drug is constantly increasing over time. Unlike in vivo drug absorption, there's no systemic distribution or elimination phase, so you don't see a peak followed by a decrease. This is a compilation of all of the negative control data for each model drug. So each bar represents 1 drug, presented as mean plus or minus standard deviation. As you can see, the permeation of each of the model drugs without excipients was very consistent across experiments. And this data includes experiments conducted over a total of about 6 months. The positive control, which was 0.6 mg per mil SLS, always produced a robust increase in the permeation of all of the model drugs. So look at the magnitude of the PAP values here, between 40 and 60 or so, compared with the negative controls, where the class 3 drugs had PAP values less than 1 and minoxidil was 9. So the PAP is much higher in the positive controls, and you also lose the discrimination between the class 3 drugs and the class 1 drug minoxidil. So this question was actually pretty challenging. How to identify effects of excipients on permeation? So we used the criterion of 2 standard deviations above and below the mean, which encompasses about 95% of the values in a set of data to identify potential meaningful effects of excipient treatments. So in this illustration, the error bars represent two standard deviations. And so if either of the means, the negative control or the test, is within two standard deviations of the other mean, indicated by the error bars, that's two standard deviations, then they're not different. They both have to be outside of those ranges, not be overlapped by the two standard deviations. So in this treatment, both are overlapped by two standard deviations of the other mean. In this case, the negative control is overlapped by two standard deviations around the test mean. In this one, the test mean is overlapped by two standard deviations around the negative control. And in this one, neither mean is overlapped by two standard deviations around the other mean. And this is the only case that we would call different. So this is an example of an excipient, cross-Carmelis sodium, with no effects on model drug permeation. The time courses of one of the model drugs, atenolol, are shown on the right, and that data was used to calculate the AUC of cumulative receiver concentration versus time, which is shown on the left for all of the model drugs. PAP was also calculated, but I'm not showing that here, we're just showing the AUC. About half of the excipients, 7 out of the 15 that we tested, looked like this, with no effects on the permeation of any of the model drugs. SLS is the one excipient that consistently increased the permeation of all of the model drugs, indicated by the asterisks. Acyclovir was not flagged as different because of the magnitude of the standard deviations, but was clearly trending higher than the control. And I want to point out the modest increases in AUC with increasing SLS concentration. Although SLS is a detergent, at these concentrations, it's not destroying the integrity of the cell monolayers. However, if you look at the time course graph, you can see that when we monitor permeation out to 120 minutes, SLS at the highest concentration, 0.3 mg per mil, eventually causes a large increase in model drug permeation at longer time points, probably indicating a loss of cell monolayer integrity. Because of that, so we can look at effects that are not due to cytotoxicity, the bar graph on the left is based on model drug permeation out to 60 minutes only. The fact that SLS up to 0.3 mg per mil was well tolerated, at least for 60 minutes, is very different from results that we and others have published on SLS and Keiko-2 cells in the conventional static horizontal transwell format, where monolayer integrity was lost even at 0.1 mg per mil. This is a summary of the results of this phase of the project. So 7 excipients, hydroxypropyl methylcellulose at 2 viscosities, microcrystalline cellulose, cross-Carmelis sodium, talc, mannitol, and silicon dioxide had no effects on the permeation of any of the model drugs. Another 6, povidone, magnesium stearate, lactose, calcium phosphate, pre-gelatinized starch, and PEG 400, had some effect, either an increase or a decrease in permeation on only 1 or 2 of the model drugs. One excipient, sorbitol, was inconsistent because it affected the permeation of all of the model drugs, but the effects were in different directions in 2 separate experiments. Now, sorbitol is known to reduce drug absorption in vivo by decreasing intestinal transit time, but it's not known if it affects the intrinsic permeability of drugs. The only excipient that produced dose-dependent effects on the permeation of all of the model drugs was SLS, which is not surprising because it's known to be a permeation enhancer. So to summarize, first of all, Keiko-2 cell monolayers in IDOS were less sensitive to excipients than in transwells, which was one of our hypotheses going into this project. Second, most of the excipients that we tested had little or no effect on the permeation of the class 3 model drugs. Keeping in mind the disclaimer that I presented earlier, that the opinions expressed here are mine and do not reflect the views or policies of the FDA, the results to date suggest that it may be possible to safely expand biowaivers for BCS class 3 drugs to a wider range for some excipients. But a lot more work remains to be done before we get to that point. So in year 2 of the project, we're flipping the script. Instead of testing one excipient at a time with a cassette of model drugs, we're testing one model drug at a time with multiple excipients to establish an in vitro, in vivo correlation, and we're looking at both dissolution and permeation. And then the plan is to use that in vitro data to predict in vivo performance in combination with a Pbpk model. I want to thank you for your time and attention, and I want to acknowledge my colleagues at Absorption Systems, especially Yuhua Huang, who did all of the bioanalysis, Nicole Spivey, who did all of the biology, and the core team at the FDA that we interact with on a regular basis. Thank you very much.
Good morning. First, I want to thank the organizing committee for inviting me to present in this workshop. The topic I'm going to present today is about whether we are ready to apply oral Pbpk modeling for BE determination. I have been working for Apotex for over 30 years. Apotex is a multinational generic company. As you know, generics can significantly reduce the cost of medicines, but they first need to be demonstrated to perform just as well as the brand products before being available to patients or before physicians have the confidence of prescribing it. On the regulatory side, the regulators have to ensure proper testing of generic drugs be done. However, significant saving can only be passed on to customers or patients if generics are not overburdened with unnecessary studies or requirements. So it's important for regulators to foster this environment. To ensure equal in vivo performance, generics need to be bioequivalent to the brand products. To demonstrate bioequivalent, the main in vivo study requirements are single dose, fasted, and/or fed comparative bioavailability studies. In general, the current BE methodology works well. However, BE studies are not cheap. They could cost few thousands to over $1 million USD, depending on the sample size and the study duration. They could take 3 or more months to complete, depending on the complexity of the study design. So any waiver of BE studies would provide significant cost and time savings. And the savings would be much greater if clinical endpoint studies are involved. Therefore, we embrace the application of Pbpk modeling for BE determination to reduce the burden of conducting BE studies. One common application of Pbpk modeling for the generic industry is to use it to develop clinically relevant dissolution specifications. Typically, the following steps are involved. The first step is to assess the clinical relevance of the dissolution test. The second step is then to identify physiologically relevant parameters contributing to consistent on-target AUC or Cmax tests to reference ratios. After the Pbpk model has been established, we then use it to simulate the PK profiles for the pivotal bio batches and the hypothetical lower bound and upper bound batches. After the simulation, we then apply bioequivalence assessment on the simulation data for the lower bound and the upper bound batches. Based on the assessment, we can then establish the clinically relevant dissolution specification. With this establishment, we can allow Biowaiver for post-approval changes in formulation of an approved drug product. In the next few slides, I'm going to use an example from the Apotex file to demonstrate this application. This example is based on our experience with a control-release product with a highly soluble drug. This figure displays the dissolution profiles of 4 batches of the CR product and 2 batches of the brand products. In addition, the TR ratio of Cmax from the corresponding biostudies will also displayed in this figure. Batches 179, 130, and 213 were batches with fast, slow, and medium dissolution, and their data were used for the Pbpk model development. Batch 135 was made with a different formulation approach, and it served as the external batch for the model validation. As you can see, this figure demonstrates a strong rank order correlation between the dissolution rate of the batches of the CR product used for the model development and the corresponding TR ratio of Cmax observed from the bio studies. This strong correlation supports the clinical relevance of the dissolution test. With the appropriate dissolution and PK data being obtained, the next step is to identify physiologically relevant parameters that were needed to develop the Pbpk models using GastroPlus. This table displays the list of the relevant parameters. They include the partition coefficient, the pKa of the drug obtained from the PubChem database. The solubility of the drug was obtained from the in-house data. The human effective permeability was derived from the Cato-2 permeability data from the literature. The gut physiology absorption scale factors were based on the default values of GastroPlus. The first pass effect and the fashion unbound were obtained from the literature. Now, it should be noted that some PK parameter values were estimated based on the published data for the immediate release product of the drug. They include the volume of distribution, the clearance, and the intercompartmental constants. After assigning the relevant parameter values, the performance of the Pbpk model was assessed by simulating the PK profile of the IR product and compare that to the literature published concentration time data of the IR product. In this figure, the square symbol represents the literature data of the IR product, while the blue line represents the PK profile simulated by the Pbpk model. As you can see, based on the initial parameter values, the model fits the PK profile of the IR product well. The next step is to develop the Pbpk model for the CR product. This was accomplished by using the dissolution and bio data of the fast, target, and slow batches. In addition, some PK parameter values were adjusted from their initial value that were mentioned in the previous slides. The appropriateness of the Pbpk model for the CR product was tested by simulating the PK profile of the external batch 135 and compare that to the actual bio data. Now, as shown in this figure, the blue curve represents the simulated PK profile of batch 135. As you can see, the profile fits the actual bio data of batch 135 reasonably well. To further validate the established Pbpk models for the CR product, The accuracy of the simulation was calculated for all batches. The results are summarized in the table below with the % prediction error calculated for Cmax and AUC from 0 to T. These are the values for the 3 batches that were used for the model development. And these values were the one obtained for the external batch 135. This column is the mean absolute % PE based on the 3 batches. And the last column is the mean absolute % PE after including the % PE values of the external batch 135. Based on these values, the predictability of the Pbpk model can be considered adequate, as the % PE for both PK parameters of all the batches did not exceed 15%, and the mean absolute % PE was less than 10% for both parameters. Now, after the model has been validated, the next step is to use the model to justify the dissolution test acceptance criteria for future batches. Based on the dissolution profile of batch 213 that has been shown to be bioequivalent to the reference product before, 2 hypothetical dissolution profiles representing the so-called lower bound and the upper bound batches that are believed to still be bioequivalent to the reference products were proposed. The idea here is to simulate the boundary for the dissolution specification. Now, this table displays the 2 hypothetical profiles for the lower and the upper bound batches. Now, in practice, a number of attempts are usually needed to come up with these final hypothetical profiles. The next step is to assess BE of the lower bound and the upper bound batches to make sure that they would indeed produce PK profiles that are bioequivalent to the reference product. This was accomplished by using the established Pbpk model to simulate the PK profile of the lower bound and the upper bound batches for the same number of subjects as in the pivotal BE study for batch 213. Now, to generate random variation of data, the gastro plus default variabilities was incorporated into the simulation. For example, we used 20% CV for the first pass effect, 40% CV for the systemic clearance, 20% CV for the volume distribution, and 10% CV for the body weight. Now, for each simulated PK profile, we determined the Cmax and AUCT value. For the BE assessment, we then determined the ratio of the geometric means between the hypothetical batches and the reference product. This table shows the geometric mean of the actual bio data for the reference product and the simulation data for the upper and the lower bound batches. This is the column for the ratio of geometric means for the comparison of the upper bound and the reference product. And this one is for the comparison between lower bound and the reference product. As you can see, all the ratios are within 0.8 to 1.25 for both lower bound and the upper bound batches, thereby supporting BE of these 2 hypothetical batches. After confirming BE of the 2 hypothetical batches, the final step is to set the acceptance criteria for dissolution profile of future batches. This was accomplished based on the dissolution profile of the lower bound and the upper bound batches. This table shows the chosen specification times and limits. By meeting the dissolution specification limit, a waiver of BE study can be granted for future batches of the CR product. What I have just discussed is a common application of all Pbpk modeling for BE determinations. We obviously will appreciate more opportunity to apply Pbpk modeling for waiver of BE studies. This slide shows a list of possible applications of this modeling. The first possible application is to extend the Biowaiver that I have just presented to you to post-approval manufacturing changes that exceed the SUPAT Level 2 requirements. The second possible application is for Biowaiver for non-proportional formulations. Typically, BE studies are conducted on the highest strength. A waiver of bio studies can be obtained for lower strengths if they are similarly formulated and display similar dissolution as the highest strength. So could we use Pbpk modeling for demonstrating BE of non-proportionally formulated lower strength or proportional formulations but with dissimilar dissolution profile? The first possible application is for waiver of fat BE studies. BE study under fasting conditions instead of fat conditions is typically required by regulatory agencies because it has been demonstrated to be more discriminatory in detecting differences in bioavailability between formulations or products. But some agencies like FDA still require fat BE study. So could we use Pbpk modeling for simulating fat conditions to assess BE instead of actually conducting the fat BE study? The final possible application is for biowaiver of BCS class-free drugs. For BCS class-free drugs, all of the excipients should be qualitatively the same and quantitatively similar to obtain a waiver of BE studies. But could we use Pbpk modeling to justify biowaiver for Q1, Q2 non-compliant formulation? Before Pbpk modeling can be more widely used, however, we believe there are challenges that need to be overcome. First challenge is that many people, including regulatory authorities and clinicians, simply do not trust modeling. They think that Pbpk modeling is too complicated to understand. For example, the presence of black boxes that represent various GI compartments involved during drug absorption. Second concern they may have is that estimation of parameter values or results is done by simulation. Unlike computer fitting, simulation results may be performer dependent, as different performers can stop the simulation at different points. So they may suspect that the simulation results could be manipulated to produce favorable outcome to the performers. The last concern they may have is that different softwares used may result in different outcome for the same set of data, because assumptions of certain physiological parameters may be different between softwares. The second challenge is that simulation of GI conditions, like the fat state with high fat, high calorie food, is difficult. For example, it can be difficult to simulate the interaction between food and drug particles. Also, the impact of food on GI motility of drug could be highly variable. The last challenge we believe exists is that model validation process could be demanding, such that extensive validation could require a large amount of in vivo data that may negate the cost and time saving. I look forward to engage into further discussion on overcoming these challenges or more in the panel discussion. Thank you for your attention.
Hi everyone, my name is Zhang, and it's my pleasure to be the moderator for the panel discussion together with Dr. Tycho Heimbach. So in addition to the four speakers who have given us a very excellent presentation, so we have additional 4 members who will be joined our today's panel discussion. Dr. Amitava Mitra is the Director of Clinical Pharmacology and Pharmacometrics in Janssen R&D, where he supports global drug development program. Dr. James Polli is a Professor of Pharmaceutical Science and Roppk Knoxville Endowed Professor in Industry, Pharmacy, and Pharmaceutics, University of Maryland. His research interest is oral drug absorption and formulations involving laboratory and clinical research. Dr. Liang Zhao is the Director of the Division of Quantitative Method and Modeling, Office Research and Standards, OGD. Dr. Zhang is currently serving as a Senior Advisor for Oral Drug Product in Office of Research and Standards. She is a subject matter expert in overseeing publications of product-specific guidances and leads multiple research projects to address regulatory scientific issues, including BCS3 Biowaivers. So next, I would like to pass the podium to Tycho and start our panel discussion. Thank you.
Yes, good morning, everyone. It's great to be part of this workshop, and we will immediately go to some questions which we have pre-prepared with input from various audience members and the experts. So the first question is, what are the common reasons for non-acceptance of Pbpk-based biowaivers? Question is perhaps best or partially addressed by Drs. Fang, Wu, and Amitava Mitra.
Thank you, Tycho, for the question. The common reasons we see for non-acceptance of Pbpk-based biowaivers or risk assessment include model is not sufficiently validated for its intended purpose. And also we see the model inputs. This include dissolution or solubility. They are not biorelevant or biopredictive. And some cases also the deficiency or limitation of the modeling include the lack of justification for input model parameters. And also lack of non-BE batch to challenge the model and increase our confidence on the model. So I generally put this couple of the common deficiencies here. And we also have some publications listing the deficiencies we saw in the Pbpk model during the review in our recent publication in AAPS journal. Thank you.
Thank you, Fang. Hello, everyone. This is Amitava Mitra from Janssen. So just to tag on to that, onto for Fang's first point there, at least from my experience, my perspective, and just talking to others, the most common feedback for non-acceptance is model not being fit for purpose, or the model is on the same vein is that the model is not verified with enough different formulation variations or variants. And this is particularly important for if you're going for a biowaiver or a specification justification based on Pbpk, that there is sufficient variance of the formulations being tested. But this is also problematic, especially the point of using and having a non-BE batch, which is not always either technically feasible or even logistically possible to generate one. And the other one, again, one of the main points that at least from my experience I've seen is not enough clinical data included in either the construction or verification or qualification of the model. And one thing that has come up repeatedly is a non-availability of IV data, human IV data, that is, for the model construction, which can also be a double-edged sword. It is always ideal to have one, but sometimes it may not be feasible or even either technically feasible or logistically difficult to generate the data. So I'll just stop there. Thank you.
Thank you for these insights. Certainly nobody likes to have their Pbpk model downright rejected. So we always appreciate when we get helpful comments from FDA or other global reviewers on how the model could be made acceptable. Our second question has to do with the drug product specification setting and virtual bioequivalence, and what should be used as reference. So the question reads, if PBPK modeling is used to justify the ranges of drug product specifications using virtual bioequivalence, what should be used as the reference? Is it the profile of the upper bound, the target profile of the test product, or is it the target profile of the RLD? I think this question, I'm going to send it to Dr. Kim Rains or Amitava Mitra.
Thank you, Tycho. I can start off. So the reference selection is going to be case specific, and going back to what Amitava said in the last response, it's really the fit for purpose. So it depends what it's being used for. So if these are to support a post-approval change, the reference may be your target profile of your drug product, but if it's for bridging or some other issues, it may be related back to the reference. So again, as Tycho previously said, we really want to see these as successful cases. So if there are questions about, what your reference should be, I would really take those opportunities to contact the agency for us to have that dialogue and those questions. You just want to also consider, building up safe space or other approaches by these virtual BE, even if you can't specifically justify your specification, there is a lot of information that can be leveraged by these models that you can use for post-approval changes and in your future space. So just leveraging any data that you may have for different formulations may also be useful.
Thanks, Kim. I totally agree with Kim's response there. I think the model being fit for purpose is absolutely necessary, and especially when you're talking about justification or specification, it's important that you have enough variants being tested and appropriately input in the model formulation variants, that is. And the one thing just to add to that would be, as was showed in the last talk, I think very nicely demonstrated, was have a range of virtual dissolution profiles being tested and set here. The BE bounds based on virtual is again a critical thing to do for this specification justifications. Thank you.
Passing the baton on to Hongling.
Hi. Our next question is for Chris and James. So in James' presentation, we can see the in vitro dissolution absorption system at DAS was used to evaluate the impact of excipients on permeability. So our question is like, are these in vitro results translatable to in vivo performance? More importantly, is how the sponsor can use this system to demonstrate that a given excipient does not impact the permeability, for example, to use to to support a Biowaver.
I'll start, Jim. So what we've found is that in our model, in the ZyDOS model with Keiko-2 cells, it's less sensitive to excipients than Keiko-2 cells in the standard Transwell model. But the FDA has done some modeling in year 2 of this project and found that it still overpredicts the effects of some excipients, SLS, I think in particular. So, and I think that's probably not surprising because it's still a static model. So in vivo, the excipients and the drug are moving down the intestine, and each ring of cells enterocytes is exposed only for a brief time. But because these are class 3 drugs, so they have low permeability, in vitro, we have to expose them to the Keiko-2 cells long enough so that we can measure the concentration on the receiver side. And that takes 30 to 60 minutes, or, in this study, we've gone out to 120 minutes. So it's kind of a worst case scenario. It's like looking at a window of cells at some point in the intestine where everything just stops for that long. So it's non-physiological in that way, but it's better than anything else we've had so far in vitro. And I think if in that model we show that there's no effect of an excipient on permeation, I think that's translatable to in vivo.
Yeah, Chris, I would agree with that. I'm just as you were speaking, Chris, I'm just going back in time, many years ago when lots of people were doing these experiments, including Keiko-2 experiments. Probably fewer people today than yesteryear, at least in academics. I think the reason why it became such a popular method is because it is a very sensitive method, right? It really punishes poorly permeable compounds. And I think in the same way, it's very sensitive in vitro to potential excipient effects. In general, as evidence from the literature, that it's overly sensitive. And when one compares what's done in vitro versus in vivo, the evidence is pretty overwhelming that, at least for common excipients, cell culture is very sensitive, and arguably that's a very good thing. I view that as a good property.
Thank you, Chris and James. Yeah, I think there's the sensitivity in vitro is definitely much higher than the in vivo performance. I think that's a good part. So I also saw, Chris, your presentation. So for future research, they're going to do like a multiple excipients together to see if any interactions in vitro for different excipients. Yeah, I think there's some gap, but it's very promising. Thank you for the very nice answer. So our next question is for Chung. In today's presentation, we heard more than one presentation mentioned biorelevance dissolution. So our question is, have you explored the use of this biorelevance dissolution data to support your Pbpk modeling and show how this biopredictivity is established for dissolution and the permeability data?
The initial release profiles in my presentation were generated as per the QC test conditions using USP apparatus 1 in the phosphate bubble pH 7.5 with sodium chloride. Now, as you know, the typical expectation is to establish the correlation using the QC media dissolution, so that the risk of bioinequivalence would be routinely controlled. In my example, the drug involved is a highly soluble drug, so the in vivo rate of drug release is formulation dependent, as indicated by the rank correlation of the dissolution rate and the Cmax data from the bio study. It should be noted that the traditional level A IVIVC was also established, but due to time limitation, I did not show it in my presentation. As a result of the high solubility, we did not find the need of using biorelevant dissolution media. With respect to the permeability, the human effective permeability parameter used in the Pbpk modeling was based on the literature reported KO2 permeability data for the drug. I hope I answered the question.
Yes, thank you. Then we have another question, and this question is for Dr. Zhang from FDA. What are the considerations for validating a Pbpk absorption model to support biowaivers? And perhaps you can also talk about model tweaking, for the lack of a better term, and maybe talk about top-down aspects, or whatever your thoughts are, validating Pbpk absorption modeling. Do we have Dr. Zhang?
Hey, yeah, you're on mute.
Sorry, I was double muted. Can you hear me now?
Yes.
Okay, thank you, Tycho. So when we talk about validation of the Pbpk model, we really want to see if the model fits the intended purpose, or so we want our model can predict, provided the the accurate and efficient prediction of our in vivo performance. So first of all, I really strongly recommend the sponsor to refer the details for our guidance for industry on PBPK modeling, as Kim mentioned in her presentation. And so the validation of the modeling, the amount of and the type of the data needed for model validation could be varying depending upon the clinical risk and the intended use. So I would like to highlight a couple points. First of all, during the modeling validation, we strongly recommend independent data sets which were not used for established model be used to evaluate the established PBPK modeling to predict the in vivo performance. Second, I would like to highlight as sponsor should include both, should include the not only the bioavailability study data from the batches you already demonstrated bioequivalence under testing. You should also challenge your model with those field bioequivalence or bioavailability data to support the robustness of your PBPK modeling. Third, I would strongly want to highlight is strongly recommend that the sponsor can perform their validation by obtained from different doses and batches with different release rate to support establish your in vitro in vivo relationship. That's pretty much it from me. Thank you.
Thank you for this detailed answer, Dr. Tsang. Very helpful. And we have a follow-on question. This question goes to Dr. Liang, Zhao, and Yu-Chung Tsang. This has to do with what can the agency do to further promote and encourage the use of PBPK in regards to model use in the for both innovator companies and generic companies.
Yeah, probably I can open up, then Yu-Chung can follow. So basically, I just want to thank all the speakers and audiences who has the interest and to share the work with us and pose questions and challenge the status quo. I think modeling is here to generate value rather than pose another burden to the industry. In this regard, we share the common interest. Think from FDA perspective, industry's interest is especially for modelers like us, is also our interest. We together want to reduce regulatory burden and to make the high quality product accessible to the general public. In that opinion, we have been internally discussing, the model verification validation could pose, as some of the presenters mentioned, could pose even a higher burden than just actually conducting the study. We really want to running into that scenario. So certainly, we are promoting, the modeling practice as more purpose, both purpose and the risk-based. Risk-based meaning, you can win a Nobel Prize by figuring out a physiological parameter. But for PBPK models, we cannot, the truth that we cannot get all the parameters figured out and have the model to fit into all situations. We really don't want to run into that scenario. It should be purpose-based and risk-based. For example, if a drug that has high solubility and is not pH dependent, should we evaluate the excipient effect on the GI tract pH level? I don't think so, even though it's a good scientific subject for other drugs, but it may not be important for that particular drug. If a drug have a high permeability and we do not know the exact coefficient for permeability, but we know it's sufficiently high, do we need to figure out that one? Certainly we can circumvent the issue by conducting sensitivity analysis. What would be the worst case scenario? What would be the worst case excipient effect? Would that pose a risk for bioinequivalence? I think we are reasonable modelers, and if the industry can think along, with more creative, implementable approaches with the FDA scientists, we will form very good, healthy, productive ecosystem to tackle all the difficult problems. I also share my appreciation to Chung. He listed 4 areas, especially for Pbpk absorption modeling, that industry do want to have modeling impact, like a Biowaiver for post-approval changes, and a Biowaiver for non-proportional formulation, and a waiver for Fat B studies, which will be covered in the next session. Don't want to speak too much on that. And a Biowaiver BCS class 3 drugs. I think we are internally aligned with all those industry need. We just need a time and a more scientific effort into those issues. Thank you.
Thank you, Dr. Zhao. And Dr. Tsang, I know you had a slide there at the end, so I'm sure you have something to say.
Well, I definitely concur with Liang that the amount of data required to validate the Pbpk model should not negate the time and cost saving that can result from the use of the Pbpk models. I guess in addition to that, building a database with some model input parameters for physical chemical properties of drug substances, PK parameters on absorption and systemic disposition of the drugs, effects of excipients on absorption of drug, etc., that can be shared will be helpful in minimizing differences in modeling and simulation outcome for the same drug product caused by different performance or software. This could decrease doubt on the reliability of PK modeling and simulation. Also, I guess more education of clinicians and other regulatory authorities on how PK modeling is developed and validated, as well as why certain assumptions, such as those mentioned by Dr. Jesko this morning, that should not significantly affect the outcome, could also increase the acceptability of Pbpk modeling. I think those are things that could certainly increase the use of Pbpk modeling from a generic perspective. Thank you.
Thank you, everyone. I am very cognizant of the time here. In terms of the wrap-up, we could say a lot of things. We had a lot of case examples, decision trees, relevant in vitro data, in vivo data, the guidances. To me, what I'm walking away with, we should challenge the FDA, we should challenge ourselves, and as Dr. Zhang said, we should also challenge the model. So I'm going to pass it back on to the organizers, and thank you.
Thanks, Tycho. We're a little bit behind schedule, but the session 2 will begin at the top of the hour at 11 o'clock Eastern time. Have a good coffee break.
Thank you.